I'm really excited to speak to this cool audience of people, and I'm really excited to remind everyone that microbes are people too. I know that because I share a lot of properties with microbes. In fact, we know some of the same things, such as the fact that life is very stressful. So whether you're standing in front of a bunch of people you don't know giving a talk, or if you are just a single cell floating around in a menacing environment, you have to learn how to adapt. And this is something the cells have to face. So you're constantly faced with all sorts of inputs that need to be sensed. And the way that cells do this is a vast diversity of ways, which we typically just kind of call as a single processing step that then results in some sort of output. Now, this signal processing we usually treat as a black box, and I, as a biologist in recovery, learned in my undergrad that this was the answer, that this was signal processing, is that the cell is full of stuff. This stuff is usually protein and RNA, and if you squint hard enough at it, you can see all sorts of interactions, and that's the answer to signal processing. For me, that's not really a sufficient answer. So what I've done throughout my PhD and now throughout my postdoc is to try to take these sort of cartoons and give them a very rigorous mathematical treatment to try to understand in a predictive way how cells sense their environment and respond. And the ultimate response, if you're a microbe, is the ability to rapidly grow. So what I want to try to talk about today, I'll get through most of it, is trying to understand how cells coordinate this vast array of different processes to not only grow quickly, but to be able to grow at all, and then also just what the fundamental physical limits are that define how fast growth can be. So rather than jumping straight into data, what I would like to do is I would like to have everyone suspend their disbelief here, and we're going to try to think up a self-replicating system from scratch. So the way that I like to think of these self-replicating systems is that we imagine that we have some well-mixed environment full of a clonal population of cells. They're all genetically identical. There's no standing variation. They behave identically. And they're growing in an environment that is incredibly well-defined. I'm going to then commit the ultimate sin and say that cells don't even exist, and all that we really have to care about is the total accumulation of biomass as a function of time. And rather than keeping track of all the individual types of proteins and RNAs, I'm going to make the approximation that this biomass is mostly protein, and we can categorize these proteins into one of three different classes. Either they're ribosomal, they're metabolic, or they're all the other stuff that cells need to do to live and prosper. So this cartoon kind of captures what I think of as in terms of a self-replicating system, but we can make it a little bit more quantitative by turning this into a sort of mass flow diagram. So if I wanted to try to understand what defines the rate of growth of this system, I would want to be able to track how I have nutrients around an environment that then get passed through all the metabolic machinery of this system, ultimately becoming precursor molecules, as I'm going to call them. You can think of them as charged tRNAs. And those tRNAs or precursors then pass through the process of translation to make new protein. Now, this is where the problem lies, which is how does, and I'm going to anthropomorphize a bit here, how does a cell decide how to invest these precursors to either make ribosomes or make metabolic proteins or make the other proteins? Because if you make more ribosomal proteins, that has to be made up somewhere. You have to make fewer metabolic proteins, for example. So with this type of mass flow diagram, we can try to mathematize each one of these arrows. And I'm going to just show you what these coupled ordinary differential equations look like without going into the details of their derivation. But what I want to point out is that there's really just kind of two tiers that we have to worry about. This top box describes how biomass or the precursor concentrations accumulate with time. And this bottom box depicts the process of allocation, or how cells decide what goes to be ribosomal, which I'm always going to denote as this phi R, or phi RB, and then what becomes metabolic, which I'm always going to denote as phi MB. So if we have this handful of differential equations, we can numerically integrate them, and we'll be relieved to see that, hey, we kind of get this mixed batch culture growth out. That seems good. That seems like we're capturing something here. So this makes some intuitive sense that if you look on this far left-hand side, we have the concentration of nutrients in the environment, which ultimately get consumed and depleted as the cells grow. This results in an exponential growth of biomass until the nutrients are completely depleted. And then if we look at the concentration of precursors as a function of time, you see that we enter this plateau region where the concentration isn't really changing, and that is what we're going to call the steady state regime. So we can take this handful of equations and then interrogate the steady state regime to see if we can make some concrete predictions out of here. And again, skipping through the details of the derivation, we can come up with closed form analytical solutions that describe the translation rate, which I'm going to denote with gamma here. So this is the rate at which peptide bonds are being formed per ribosome per unit time. And then we have the steady state growth rate. So that describes the rate at which the entire system is growing. And we can, as one of our control variables here, we can tune how this allocation towards making new ribosomes affects both of these properties. And if you look at the right-hand plot, along the x-axis, we're either going from making no new ribosomes at all or only making ribosomes. And it makes sense that in both of those cases, the growth rate is 0. In the case where you don't have any ribosomes, nothing to make new biomass. In the case where you don't have only ribosomes, well, then there's no stuff to metabolize the nutrients. So that implies that in between here, there's a sweet spot. And this changes with concentration as well. But in between these two extrema, there is a sweet spot, or this peak, where the growth rate seems to be maximized. So we can analytically compute where that position should be, and again, focusing on our control variable, which is this allocation towards ribosomes. So this now gives us some predictive things to look at the steady state regime of growth. And if you've noticed, up to this point, I haven't mentioned a single organism's name, because this in principle should be kind of organism agnostic, so long as you have protein limiting growth as the key thing. But what we can do is we can go to our dear old friend E. coli, and we know a lot of the parameters that are involved in this model. So we can plug in some things to make some predictions of how these two different parameters should scale with the growth rate, for example. So if I wanted, what I could do is I could go into the lab, I could use some transcriptomics, or I could do just protein quantification to measure these things, or I could do even better and then go to the past 65 years of research that's been done. And we can go through all of the literature. These are all the literature sources that I was able to find that could actually measure these things in E. coli. And this is covering about 55 years across different groups, different generations of scientists. And once we had these predictions in place, we were then able to plot the data. And what we see is a pretty remarkable agreement here. So we could argue a little bit about what's going on at slow growth regimes in terms of the allocation towards ribosomes, but largely I would argue that this does a pretty good job of describing both the ribosomal content and the translation rates. But to me, this sort of raises more questions than answers. First is mechanistically, how do cells optimize their growth rate? I think of growth rate kind of as a output. I don't think of it as a measurable quantity that can be directly tuned biologically. And then also evolutionarily, why would cells even optimize for steady state growth in the first place? That is a pretty contrived regime. That's not something you frequently encounter in the gut, for example, where nutrients are changing all the time. So in this recent work, what we did is we tried to get at this first question, and we asked mechanistically, what is a way that cells could actually optimize their growth rate? And one way that they can come up to do this is by being able to simultaneously measure the processes of metabolism and the processes of translation together. So in steady state growth, by definition, these two processes, their fluxes need to be in balance, right? That's the only way that you get this steady state regime. But there is a sweet spot in which this allocation towards ribosomes will not only will these two fluxes be balanced, but they will both be mutually maximized, meaning any increase or decrease in this allocation towards ribosomes leads in a net decrease in one of the two fluxes compared to a different value. So how can we mathematize this kind of logic? One way we can do this is we can, rather than talking about one collective pool of precursors, what we can do is we can separate them. And we could say, let's consider that we have charged and uncharged tRNA species. Now, the charged tRNA species come through metabolism, and that's going to be, and their rate is dependent on how many uncharged tRNAs you have around. And the uncharged pool comes from the process of translation, making new uncharged tRNAs, and transcription as well, and then those will get charged through metabolism. So to identify this kind of sweet spot denoted here by this red vertical dashed line, we call this thing a flux-parity regulatory motif to be, catchy and cool. And if we were to again draw a sort of mass flow diagram, If you have nutrients coming in this side, then this is the system that we have. So we can do the same thing, and we can mathematize these cartoons. And when we integrate them, what we get out of this is something, at least to me, that was pretty remarkable, is that for a wide range of parameter regimes, we were able to exactly recover what the optimal scenario is. So that indicates that by being able to measure these two different pools of precursors, you can actually do a pretty good job of predicting, or you're able to enforce the sort of optimal behavior to come out of it. Now, what's nice is that because the model is dynamic in its construction, we can actually start to test this thing out of the steady state regime. So one example of this is we can think of nutrient shifts, where we go from some poor substrate, and all of a sudden there's an inflow of new nutrients, consider it as an inflow of nutrients. So the cell has to be able to adapt to this by it can afford fewer metabolic proteins because the nutrients are rich now, and it can then want to make more ribosomes such that the growth rate can increase. So if we just plug in some parameters that we have for E. coli into this model, and we numerically integrate it and allow it to perform, we see that we do a really good job here of being able to predict how the instantaneous growth rate changes at the point where you have an inflow of rich nutrients. So we're able to make some predictions about the scale of adaptation. So the way that I think about cellular decision-making now in the context of growth regulation is kind of in two steps. There's one, the first step is that the cell has to kind of decide what to express. So evolutionarily, for example, E. coli in the gut is being exposed to all different types of nutrients coming in, and that's led to some sort of evolutionary beneficial spread of metabolic proteins to express. So you may express a transporter for a sugar that's not around, but maybe it'll be around in some cases. So there's going to be a spread of things that are expressed. The other level of regulation is then, okay, well, if I have what that spread is, how do I properly allocate my resources to get there? Okay. And in the last 30 seconds that I am going to take here is that I'm really curious how else this is achieved in the wild. So this is a phylogenetic tree of bacteria, and I'm going to now highlight in a red circle everywhere that we've studied resource allocation. The world is wild out there, and we're still kind of figuring out E. coli operates, and I'm really curious to see where this goes.